Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
What drives tumor growth? Is it a few rogue cells imposing their will upon healthy tissue, or diseased tissue bringing out the worst in otherwise peaceable cells? Or is it a back and forth, a dialogue between the two? According to a new study, it may be the latter, at least when it comes to the progression of one common skin cancer. Researchers found that a single mutated gene in an otherwise healthy stem cell can kick off an increasingly deviant feedback loop of miscommunication between the cancerous stem cell and its surrounding tissue, fueling the development of a malignant tumor. The findings suggest that many of the mutations in cancer may simply be setting in stone a path already forged by the tumor stem cell's aberrant dialogue with its surroundings. If these results, published in Nature, prove broadly applicable, the findings could pave the way for novel approaches to treating a range of cancers. Our study shows that there is crosstalk between the microenvironment and the stem cells in tumors. They communicate with each other and create a loop of tumor-promoting factors. Spotlight on squamous cell carcinoma. At the heart of almost every tumor is a small subset of cancer stem cells. Resistant to chemotherapy and immunotherapy, these malignant seeds are the cells responsible for keeping the tumor alive and are key players in the process that turns benign growths into metastatic disease. And behind many cancer stem cells, including those of skin, pancreatic, lung, and colorectal cancers, is a ROS gene that, when mutated, allows tissue stem cells to ignore normal environmental signals and deviate from their natural course, promoting out-of-control tissue growth. To better understand the finer points of that interaction, Yuan and colleagues turned their attention to squamous cell carcinoma, a skin cancer linked to ROS mutations. The team started off by inducing mutant HRAS, the member of the ROS family most common in skin cancers, in individual skin stem cells, and monitoring how the cancerous stem cells interacted with the surrounding tissue. Over time, the dialogue between the cancer stem cell and its microenvironment became more and more aberrant, Fuchs says. A hormone produced by fat cells and linked to obesity, leptin seemed out of place in non-obese mice carrying non-fatty tumors. LEPR is not expressed in normal epithelium and rarely seen in benign tumor cells. Here it showed up unexpectedly in the cancer stem cells of the advanced stage of the tumor, known as squamous cell carcinoma, SCC. Yuan used CRISPR technology to show that LEPR and leptin receptor signaling was essential for progression from the benign to malignant state. But where was the leptin coming from? Yet, the malignant tumor expressing the leptin receptor greatly benefited from the presence of leptin. The more leptin, the faster it grew. The team began to wonder whether leptin that normally circulates in the bloodstream was arriving at the tumor via the blood vessels that bring nutrients and other factors to the tumor. Through a series of experiments, they provided compelling evidence that this was the case. Moreover, they went on to demonstrate that leptin signaling within the cancer stem cells stimulated many of the pathways known to be hyperactivated in cancers, including the PI3 kinase, AKT and mTOR pathways. The team is now investigating ways to block the leptin receptors in tumors, as in doing so, It could throw a molecular monkey wrench into the vicious loop and derail the cancer. If we block this loop, which is a major pathway driving tumor progression, perhaps we can block tumor progression. Squamous cell carcinomas affect not only skin, but also esophagus, head and neck, lung and other epithelial tissues and, together, comprise the sixth most common cancer worldwide. Our study shows that one oncogenic mutation can hijack a pathway and achieve the same thing as many cumulative mutations, Yuan says. We are always looking for mutations, but we must not forget to think about how to stop signaling pathways that drive tumor growth. Xiaopeng Yuan, Catherine S. Stewart, Yihao Yang, Merv Denise Abdusalamolu, S. Martina Parigi, Tamar Y. Feinberg, Karen Tumaneng, Hansel Yang, John M. Lavorse, Lisa. Ross drives malignancy through stem cell crosstalk with the microenvironment. Nature, 2022. doi. 10.1038, S41586, 022, 05475, 6 Rockefeller University. Cancer stem cells are fueled through dialogue with their environments. Science Daily. https://ift. tt. ZNSBB5Z. Access December 1, 2022. Thanks for watching. Please subscribe my channel for more videos.